Hello, everyone. Welcome to today's webinar. I'm Jeff Huber, contributing editor at Chemical and Engineering News, and I will be moderating today's event. This webinar is titled Analysis of Nitrosamines and APIs, and is being sponsored by MacMod. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by MacMod. A company should be able to condense its mission statement into a simple phrase. MacMod's mission statement is smarter chromatography. But you may ask, just what does smarter chromatography mean? It means that when MacMod makes a recommendation to you about an HPLC or UHPLC product, it is to partner with you to offer innovative solutions to your complex problems. MacMod uses advanced, state-of-the-art analytical software to match its product portfolio with your separation needs. The company leverages its 30-plus years of experience in the LC column business, its technical expertise, and its manufacturing network to help you solve your toughest application problems. MacMod provides up-to-date and accurate technical catalogs and reports from industry-leading separation scientists to keep you informed of new technologies. And during the presentation, we will hear from Dr. Tony Edge. Dr. Edge is a site lead at Avantor Sciences, heading a team of specialist scientists in manufacturing and developing next-generation stationary phases for HPLC. He has worked in both manufacturing and also industry, having periods of employment at LGC and AstraZeneca, as well as Thermo Fisher Scientific and Agilent Technologies. In 2008, he was awarded the Desti Memorial Lecture for his contributions to innovating separation science, and the same year also won a Clinical Excellence Award from AstraZeneca. And with that, I will now hand things over to Dr. Edge.
Jeff, many thanks for the very kind introduction. Welcome everybody to today's presentation, which will be looking at the analysis of nitrosamines in active pharmaceutical ingredients. I have to start off by just acknowledging my co-worker, Matt James, who did the bulk of the experimental work. And unfortunately was not able to attend today's lecture. But hopefully I can do him proud in terms of delivering this. We've got quite a bit to go through. But before I do that, I just want to thank the organizers and MacMod in particular for giving me the opportunity to deliver this presentation. MacMod have been a trusted partner with us for over 20 years and have a real expertise in HPLC and in UHPLC. And I'm delighted to be able to kind of do this presentation in conjunction with them. So thank you very much for this opportunity. So let's move now on to the topic for today, which is the analysis of nitrosamines in APIs. What I want to do initially is just give a bit of a brief introduction to nitrosamines and why they're so important within the pharmaceutical industry. We'll go through a bit of a timeline over some of the developments that have happened over the last 5, perhaps even 3 years. And we really have seen an explosion in terms of the interest in this area since the initial finding of the nitrosamine in a drug in 2018. But I'll go into a bit more detail on that. Where I want to try and focus, though, is looking at how we took the information that the regulators supply in terms of what we need to do to optimize and develop the assay, and take that and then start to develop our own assay. We'll talk about a whole raft of things, but we'll talk about column selection, mobile phase selection, and why that is critical for getting the optimal separation and indeed ensuring that we get good detection limits. We'll also talk a little bit about the detection technique that is used. It's not UV, it's mass spectrometry, and why do we have to use mass spectrometry in this case? And from there, we'll then start to have a look at some of the data that the assay has produced, the one that Matt produced. And then finally, I wanted to try and move on to some of the more interesting areas of that method development. Using mass spectrometry can inherently have some issues that you wouldn't get with other detectors. And one of the classical ones is looking at ion suppression. And the coelution of certain solvents with our nitrosamines can cause some quite interesting artifacts. And I've got some really nice data that Matt produced that we can go through. And then I'll finally wrap everything up as we come to the end of the presentation. Okay, so what are nitrosamines? Nitrosamines are given in the structure here. And so we can see that we've got a nitroso group coupled to an amine type structure. In terms of the nature of these compounds, the nitroso group is basic in nature. And so this will create a basic charge, but it will also then mean it can accept a proton, and we end up with an NHOH at that particular group. In terms of the formation of these nitrosamines, they're typically formed by a nitrosating reaction between an amine and some form of nitrous acid. So typically this could be a secondary, tertiary, or quaternary amine, so it could be something like acetonitrile, for example. And then in terms of the nitrous acid, what has been found is that typically it's a nitrite salt in the presence of an acidic condition that causes that. And we can see the reaction at the bottom that then ends up creating the nitrosamine. One other thing to note about the structure of this is that it is feasible to rotate the NO bond, and we then can create a syn and anti isomer, if I can use some of the older terminology. And this becomes important when we have a look at the resolution that we get, because we'll start to see some split peaks that are caused by this particular isomerization that we can get. So why are we so interested in nitrosamines? So nitrosamines are classified as mutagenic compounds. They are deemed to be carcinogenic, and indeed rat studies in the 1950s have shown that a wide range of different nitrosamines do have impact in terms of that oncology effect that you get with mammals. In terms of where they can be found in a variety of different sources, ranging from cosmetics industry, rubber, meat. Indeed, if we're looking at meat, then nitrosamines in meat was a big issue in terms of Sweden, where it was deemed that something like 80% of the NDMA intake was coming from meat in Sweden. It can also be found in beer, a range of consumer goods. Because we can digest it, it then means it then ends up in our rivers and sewage plants. But more recently, it's been found in pharmaceuticals, and this is really where the focus has been. The regulatory authorities are very interested in this, and particularly if we have a look at the ICH, FDA, EMA, regulatory authorities that control the approach to analyzing drugs. So International Conference on Harmonization, Food and Drugs Administration, and European Medicines Agency, they have identified several nitrosamines that could be present in drugs and should therefore be monitored for. And I'll go through that later on. In terms of then what is causing that mutagenic response, the nitrosamines can form a diosium ion, and these are precursors to reactive carbenium, and this forms a stable adduct with DNA, and this then changes the DNA structure, i.e. we end up with a carcinogenic type reaction. And so as a consequence of this, it is very important that we are able to monitor and try and remove as much of the nitrosamines as possible. The adducts that are formed by those carbenium ions will actually be repaired by the by the body using a cellular repair mechanism, and that changes the DNA. And in changing the DNA, we then end up with the issues that we see. I'm very quickly going to go through some of the history because I really want to try and focus on the analysis of at all possible. So in the 1950s, and I briefly talked about this, Barnes and McGee discovered that nearly 300 nitrosamines had carcinogenic properties. They did a study on a wide variety of animals, but primarily rats. In the 1960s, there was concerns raised over the excessive use of nitrate in the food preservation industry that resulted in NDMA formation, specifically in bacon and some other meat products. Then in the 1970s, there was issues in terms of the link between animal diet and fish meal and the resulting formation of cancerous cells in those farm animals. The fish meal had been preserved again with sodium nitrate, clearly indicating that the use of these preservatives is not good within the mammalian population. The first recorded nitrosamines in drug product was actually in 1977 with a compound called aminophenazone. And very shortly after being notified, the German Health Authority actually recommended that this drug was withdrawn. And we can see how the NDMA in this case was being produced. So in this case, it was being produced from the drug itself. Then in the 1980s, again, if we look at some of the disinfectants, that was causing some issues. And then eventually 2018, this is when really there was a large amount of interest. NDMA, one of the nitrosamines, was found in a batch of valsartan. And I'm going to spend a little bit of time talking about that aspect and looking at how the timeline from there really has developed to where we are today. So June 2018, a batch of NDMA was detected, and this was detected in a batch of valsartan that was being manufactured in China by the Zhejiang Huawei Pharmaceuticals Company. Valsartan is a very popular prescription drug. It treats high blood pressure and heart failure, and it's a selective angiotensin II receptor blocker, so-called ARB. There was then a recall of that. Then another nitrosamine was detected in valsartan subsequent to that one. This triggered what's referred to as an Article 31, where it required an investigation by the authorities, and from there on, it was found that nitrosamines were coming up in quite a few different types of compounds, probably the most prolific of which was ranitidine, Zantac. And as a consequence of this, Zantac has been taken off the market. Now, Zantac used to be one of the biggest selling drugs on the planet, so clearly this is having a massive impact in terms of what is happening in the drug production field. And so therefore, it is incredibly important that we can actually detect these compounds during the manufacturing process at the levels that are present, so that we can avoid that then going into the drug supply process and ending up. So in terms of looking at the regulatory aspects, the FDA has given guidance, and it has done a risk assessment, and it has produced several different documentation guidance for industries that look at control of nitrosamine impurities in human drugs. What it asks for is to assess various processes for amine reagents and intermediates, and specifically nitrate salts in acidic conditions, because this is where the nitrosamines can be formed from, as I said previously. It can also be formed from a degradation of a variety of amide solvents, such as DMF, for example. There can also be impurities from different vendors, or indeed, if solvents, etc., are recycled, it is very feasible that nitrites can be formed there. Also from potable water as a possible contamination source. So the FDA has then started asking, can we start looking at these sources, potential sources, to ensure that we are no longer contaminating the drugs that we're manufacturing. In terms of which drugs are being regulated, or which nitrosamines are being regulated at the moment, then there are the list here. So here we can see 8 different nitrosamines. The FDA has put regulations associated with 7 of these. The European Medicines Agency has 8. In terms of the acceptable intake levels, and reading the literature on this, this gets quite complicated to interpret because there are slightly conflicting advice guidance within the same documentation almost. But in essence, what the FDA and EMA have come up with is acceptable limits that can be taken. And so if we look at NDMA, then for that one, that would be 96 milligrams per day. Now, if we look at the maximum daily dose and then look at the acceptable intake limit, we can then look at what is the acceptable nitrosamine content within a particular tablet formation. And what we start to find is that some of these concentrations are getting quite low. So there are a couple of examples on the bottom right-hand side, but here we can see that if we're looking at a maximum daily dose of 1,000 milligrams, then we would be looking at concentration levels that we need to detect within the tablet formulation of approaching 96 nanograms per gram. This is moving outside the range of a UV detector and now moving into the range of mass spectrometry. And the reason for these levels is because of the potential for cancer, and as governed by ICH-M 7R1. Where it gets complicated is that if you've got different mixtures of the compound, the acceptable daily limit can alter. So if you've got lots of different nitrosamines, then actually you then may have to start to look at even lower levels of detection. And what this table basically is trying to say is roughly what limits are we looking for. As we can see here, in different drug formulations, the limits are quite different, going down to 0.12 parts per million when we're looking at NMBA in iprosartan. And these are now very much, as I say, in the reign of the mass spectrometry. So what approaches have been used in order to identify these very low limits then of nitrosamines? There have been, I guess, 4 approaches that have been documented under the US Pharmacopoeia General Chapter, and these include LC high-res mass spectrometry using a PFP column, propyl PFP column, the use of GCMS, the use of LC triple quad MSMS, and also the use of GCMS-MS. And there are the analytes associated with each of those methodologies as well. So I've talked about mass spectrometry, but mass spectrometers are very expensive. They're much more expensive than UV detectors, certainly for the type of detection limits that are required here. We're not really talking about a single quad. We're talking about a triple quad or a high resolution mass spectrometry. The operational costs are a lot higher as well. For people listening who've got a mass spec, you'll know this. You'll know that it requires nitrogen to run. You typically required fairly expert technicians in order to run the mass spectrometer, and if there is a fault, it typically costs a lot more to fix than a UV detector. It also takes a lot longer to set up the detector. So with the mass spectrometer, you tune in the compounds that you're looking for. You're not relying on a single wavelength at 254, for example, that you might do for aromatic compounds. However, mass spectrometers are very sensitive detection systems and typically would have 10 to 100 times more sensitivity than the UV detector. They're also very specific, which means that the signal-to-noise ratio is very, very good and much better than you would get with other forms of detection. It's also feasible to do very fast analysis as well, because we can look at co-eluting compounds, although there may be some issues, as we'll discuss later on, either with data collection or with co-elution. So we set the scene. We're going to be using mass spectrometry, but what's the approach going to be to developing the method? So what we're going to do and what I'm going to go through is talk about the typical approach that we have at Avantor for developing LC methods. We're going to take the compounds that we're looking at and review their physicochemical properties. So specifically looking at the log-D and also the pKa, and I'll talk a little bit about those later on in the presentation. The method was developed from a LC-UV, but as you'll see, we have looked at different alternatives in terms of columns and mobile phases, but we used the initial method that we developed in order to start that method development process. Once we've got the column conditions sorted, then it's a case of tuning up the compounds and the internal standards. This was achieved using infusion and flow injection analysis in order to do that. We need internal standards because although mass spectrometry is very, very sensitive, it does have much greater variability in terms of its response than you would expect with a UV detector, for example. And so the use of an internal standard offsets for that difference that we may see. We also looked at, if you like, semi-real samples where we'd put a real drug in there, in this case valsartan, to see what impact that would have in terms of the overall analysis. Okay, so I talked about the physiochemical properties. What do I mean by that? So there are a variety of different databases that are available where you can access some very useful information that as a chromatographer will provide you a good insight into what column to use and what mobile phases to use. And what we're specifically looking at really are looking at log P, log D, in order to work out what those key physicochemical parameters are. So log P, log partition coefficient, is simply the concentration that you would observe if you had an equal mixture of octanol and water and place your compound in. So how much is in the octanol layer and how much is in the water layer? If I was talking to environmental chemists, then we'd talk about a KOW, an equilibrium constant between octanol and water. And the equilibrium between that would give us a number. The higher the number, the more likely the compound is to go into octanol, and therefore the more lipophilic or hydrophobic that compound will be. Log D, or log distribution coefficient, really relates to a charge species, and you can see that the difference between the log D calculations and the log P are that we've now got a concentration of our ionized form in water. And it's assumed that there is no ionized form in the organic solvent. That's a reasonable assumption. But again, the premise is the same. We're looking at the relative ratio of our charged former species or our former species in the octanol layer versus the aqueous layer, and that gives us a hydrophobicity. If we're looking at trying to predict what column to use, that hydrophobicity becomes very useful. So typically speaking, if we're looking at hydrophobicities where the log P or log D is greater than 0, then we might want to use reverse phase. If the log P is below -2, then we might want to use a helix separation, for example. What it also tells us is how much organic we should need. So if we were looking at separations where the log D is quite low, we won't need as much organic in order to get that compound off. It also, when we're looking at the log distributions, will give us a really good indication of the impact of pH as well. And again, I'll show some example log D plots in a few slides, which will give an insight into this. The pKa, or the equilibrium constant, for a dissociated form of a molecule, whether that be in this case A for acid, or if it was a B, it would be a base. That gives us the point at which we have an equilibrium between the dissociated and undissociated form of the molecule. And that is really useful for trying to select the correct pH. And again, I'll go into a little bit more detail on that later on. So this sets the scene. We perform the physiochemical modeling of these compounds, and we then get a series of data coming out. And so we use a program that's available on the web called Chemicalize. However, there are a range of different types of software that are available. Some very good products from Chemaxon, from ACD, Simulation Plus, and I'm sure that I've missed some, and apologies for that. But what all of these software packages do is that they will give you a good indication of what the log D is, and I've talked about why that's important, but they'll also give an indication of where the pKa is, and let's talk about a little bit more about that. So if we now look at the middle graph, we can see a plot of log D versus pH, and what we see is that the log D of this particular compound, which in this case is not a nitrosamine, it's actually a beta blocker called propranolol, we can see that it starts off at relatively low values of log D and then goes through quite a steep transition to a stable high form. If we're doing our chromatography, we really want to try and avoid that steep part of the log D pH plot. That means that we've got small changes in pH, which could be affected by temperature, could be affected by a local modification in the mobile phase caused by another compound going through at that particular point in time, changing the number of protons that are present. That is what pH is a measure of. That could impact the log distribution. So that would impact, if you like, the hydrophobicity. And that hydrophobicity we can relate directly to retention time. So we want to try and avoid the part of the curve. So we can either go at a high pH or low pH. The rest of the information on here gives us an indication of the different types of charged species that we'll be looking for. So in this case, there are different groups of that are ionizable, which can be quite useful for optimizing our pH selection. So what do the pH graphs versus log D graphs look like when we run in then our nitrosamines? And what we can see is something that's quite interesting. We can see that the bulk of these curves look pretty flat. So that would suggest that actually pH is not going to have a substantial effect. However, if we look at NMBA, it sticks out very clearly as having being very impacted by changes in the pH. So at low pH, we've got a high degree of hydrophobicity. The log D is higher, but as we increase the pH above 8 and we move towards 9, 10, then we start to get something that's very polar. And I already referred to that up to about 0, we're probably okay with reverse phase liquid chromatography for analysis. However, below -2, and the region in between is where you might look at either approach. You'd perhaps use a Hillick approach. The other thing that comes out is that from the log Ds is that the compounds are a mixture of acidic and basic moieties. It's also very evident that there's quite a broad range of log Ds, and also quite nicely they are separated. So it means that if we just do a straight hydrophobic separation, we should start to see a decent separation of these compounds. So let's see if that actually happens. So here we've got 2 chromatograms. The top one is looking at a low pH separation, and the bottom one is looking at a high pH separation. So the first thing I want to focus on is peak 2, which was our NMBA. And remember I said that changing the pH on the NMBA would be very dramatic, and that if we went up to a higher pH, we'd expect to see the compound being less hydrophobic, and as a consequence, it would have less retention. That's exactly what we're seeing. What we'll also note is that with some of the peak shapes, they don't perhaps look quite as good. Now, I need to kind of refer to the structure of nitrosamines. I talked on the first few slides that it is feasible to get isomers of the nitrosamines, and these syn and anti isomers will potentially separate under the right conditions. And so some of the peak shape that we see here, and indeed some of the split peaks, are not because we've got poor chromatography. Actually, it's completely opposite. We've got really good chromatography, and what we're now doing is separating these isomers, which are based on that rotational movement of that NO group, so that we're moving then the free electron pair to one direction and the NO group to the other direction. That is incredibly difficult to do, so we're quite lucky that we were able to achieve this on the ultra-coarse C18. particularly when we start looking at the length of that column, which is only 5 centimeters long. So the gradient conditions are given. We start off at 3% B, and the rationale for the lower organic again relates to those log distributions. We stated at the start that the log Ds were quite low, and so therefore we don't need much organic to elute these compounds off. We go up to 100% because we are very aware that when running real samples, we want to clean the column ready for the next injection. If we look at the rest of the compounds, when we change the pH, we can see that there isn't so much difference in retention time. And again, that's exactly what the log D plot saw. So in a way, this is starting to help us, using that modeling approach, starting to help us develop the method. In terms of the mass spec method, the things that we need to consider for that are that the addition of, in this case, a weak acid, formic acid, does help in terms of some of the sensitivity. We did look at electrospray, but having read a lot of articles, electrospray was suggested that this is more impacted by ion suppression. So we moved to APCI, atmospheric pressure chemical ionization, which seems to be the preferred approach when analyzing nitrosamines. These are the conditions that we set, and so for all of the transitions, a couple of points to note. The first is that the molecular masses of the bulk of these compounds are very low. And when I say very low, it's where we've got masses that start at about 100 or below. This is very difficult when we're running with LCMS. Because of all the buffers, etc., that we get within the solvent system, it's very common to have a lot of background masses that are below 100. And most or a lot of compounds will break down into masses that are below 100, so any form of impurity there. And so as a result, it can be quite difficult to get very selective mass spec transitions. And you'll see that some of the transitions are going from, if we look at NDMA, 75 to 43. So that's the first thing. The second thing to note is that we're actually looking at 2 transitions per molecule. And so for NDMA, we're looking, again as an example, looking at the molecular ion of 75 transitioning to 43, but we're also looking at what's referred to as a qualifier ion. And this becomes important in the discussion later on. We know that there is ion suppression present. Use of a second ion, a so-called qualifier ion, and making sure that that ratio between the quantitative ion and the qualifier ion is consistent will ensure that if there is ion suppression, we can pick it up and we can do something about it, i.e. start changing the chromatography. Here now is some data. So in order to get the data, we've used a SIL, a stable isotope label, as an internal standard. And we've used 4 of these for the 8 compounds that we've been analyzing. So I'm just initially going to go through the calibration curves. I'm not a big believer in R-squared. I think there's lots of ways in which you can fudge an R-squared value, but I think that you can clearly see that we're getting excellent linearity. All of the dots, the points that we get fit on the line, and when we look at the calibration range is exactly where we want it to meet the requirements of those regulatory authorities. So these are the first 2, looking at NDIPA and NDMA. If we then have a look at NMBA and NMPA, again, some really excellent data here. There are 3 replicate injections, and so because there are 3 replicate injections, and clearly there is very little spread over those 3 replicate injections, that suggests that we've got very good precision. The fact that the dots, our points are very close to the line suggests that we've got good accuracy. But I'll actually show some data later on that clearly identifies what the accuracy is and what the precision is for these. And then looking at NDA, NDPA, again, some really neat data that Matt has obtained from the lab. Really very, very impressive indeed. And another set of data, excellent data. And I feel somewhat guilty because I know how long good data like this takes to get, and to give a few seconds on air is perhaps not really reflecting the amount of effort. But perhaps we could spend a little bit more time then on the accuracy and precision data. So the accuracy is how close to the real result is it, and the precision is how repeatable can we get that? So looking at replica injections at 7 different levels, and those levels are across the range that has been specified by the regulatory authorities, then we can see that the accuracy is very, very good. Ideally, we want 100%, but we can see that the bulk of the data that Matt has produced is between 98 to 102. That's a testament not only to the mass spectrometry, but to the chromatography. Robust chromatography using those ultra-core columns will ensure that we get good data. The precision is also very, very tight. So if we look at this, we have the majority of the data points are below 3%. Again, excellent data that Matt's produced. One of the things that we wanted to have a look at is that we had a bit of time to play around and wanted to have a look at the impact that changing the weighting factor would have. And so when we do a calibration graph, we can put in different parameters, one of which relates to a weighting. So a linear fit or a partial least squares analysis, straightforward partial least squares analysis, gives us a calibration line. And even if we look at this, we can start to see that some of the data points seem to be above and some of the data points now seem to be below. Very clearly is that the case. The graph at the top right highlights this. And what we can see is that when we look at those 3 different weighting factors, linear, 1 over x, and 1 over x squared, we can start to see that just using a standard partial least squares analysis or partial regression or linear regression, we can actually see that as we start to get down to those lower concentrations of 1.33 nanograms per mil, we start to lose a large amount of accuracy. In fact, it's almost unusable at those sort of levels. However, if we go and use a 1 over x or 1 over x squared linear weighting, then actually it starts to get much, much better indeed. And another parameter that we wanted to have a look at, so again, this is just showing exactly the same thing with NDBA, another one of the nitrosamines, and we can also have a look at the impact of now changing the curve and looking at the impact of weighting. So here we've got a quadratic curve, and interestingly, using the quadratic curve, that the impact of weighting is no longer so impactful. And we can see on the top right that the response is pretty much 100% all the way. The best option for the calibration we found was actually the use of a quadratic curve coupled with a 1 over x weighting, and that's the data that we presented. So in terms of sensitivity, if we go back to some of the data that has been produced, we can see that in terms of the signal to noise ratio at the bottom standards that we're injecting, we're getting very, very significant levels of signal to noise. And this is inherent within a triple quad, even when we are looking at very low masses. So this is particularly impressive. So we're looking at signal to noise at 0.04 nanograms, which would then give us a 0.04 nanograms per mil detection limit in terms of the solution. If we're trying to calculate what that would be in a drug substance, then as a parts per billion, then we need to take into consideration another factor which is given in the slide. So we developed a method for the separation. So now we wanted to see what happened if we injected it into or spiked those nitrosamines into valsartan. First thing to note is that with some of the peaks, we can clearly see that we're starting to separate the syn and anti-conformers, the isomers, and that is a consequence of the excellent resolution capability of the ultra-core C18 column. The other thing that we noticed very early on is that we've got very good separation and a very quick analysis time. So 5 minutes, we've been able to separate all the peaks. The reason that we've got 2 peaks for each of the individual analytes is because, and the co-eluting is because we're looking at the quantifier and qualifier ion. And so they're the 2 traces from those different ions. The mobile phase is relatively simple. We've extended the dimensions to a 10 centimeter by 2.1. A 2.1 millimeter ID column is better in terms of the flow compatibility with the mass spectrometer at 0.5 mils a minute. And this injection is at 1 nanogram per mil. Clearly, we've got a lot of sensitivity here, and those signal to noise ratios that we saw earlier, absolutely we could get those. In terms of the gradient, it's a relatively simple gradient. We start off at a low composition of percentage B, and the rationale for that is that we have low log D values. This then goes up to a relatively high value. This time we're not going up to 100%. We know that the drug comes, the valsartan would come off well below 100%. So we can go up to 80%, and then we look to re-equilibrate for the last 2 to 3 minutes of the injection run. In terms of the impact of using the internal standard, again, a lot of data here, but in essence, when we look at the impact of the internal standard when we're just doing a calibration, it doesn't seem to actually have much use. It seems that actually we could use the internal standard, we might not do. But how does the internal standard fare when we've actually got matrix there, when we've got compound there? So let's look at running some standard concentration of some example nitrosamines. So in this case, we'll look at NDBA, NDMA, MNBA, and NDEA at a set concentration. So what we should see is that with or without the valsartan, we shouldn't see a difference in the response from the transition that we're getting. Now, clearly, with one of these peaks, when we look at the graph at the right-hand side, we can clearly see that we are seeing a difference in terms of the response. So this suggests very strongly that we are seeing a suppression of that signal. And so therefore, use of an internal standard would help us to overcome that issue. It's also been noted in quite a few texts that some solvent impurities, and one of which has been noted is DMF, can also cause issues in terms of suppression or interference. So we ran the LCUV and we noted that there was partial coelution with NDMA. And so what we did is look to spike NDMA at 1 nanogram per mil with DMF to assess any potential interference that we might get. And here we can see that when we're looking at the NDMA concentration, which is fixed at 1, and then looking at the DMF, we can start to see the impact that that has in this following graph. So when we look at the DMF concentration and then the calculated NDMA concentration, so the NDMA is kept constant at 1, we can see that the NDMA concentration seems to increase with increase in DMF. We can also see that the ion ratio seems to alter as well. So clearly the DMF is impacting our ability to detect in an accurate and precise manner the amount of, in this case, NDMA there. This is the reason that you really want to monitor those ion ratios. It becomes critically important. We can also do a similar experiment with that's looking at the response in terms of what we can do to overcome this. So if we look at using a different qualifier ion, then perhaps we could actually overcome this. And so here we can see that if we're looking at the qualifier ion and using that, then for NDMA, then the difference that we get in terms of the calculated concentration is not as big as when we're using the quantifier ion. And so therefore, the choice of MRM transitions becomes critically important. Looking at the calibration, again, we can look at the impact of using of the qualifier ion as opposed to the quantifier ion. And again, some interesting data there that clearly demonstrates that actually we need to be very careful when we're looking at coelution of, in this case, DMF with NDNA, of what we're actually getting. In terms of the data that comes out, we can see that the CVs are all, again, pretty tight, and the concentration, the calculated concentrations are again pretty tight when we use the right ion transition. We have started work looking at trying to separate DMF from NDMA. And in this slide, we can see how that work is progressing. So looking at the initial column that we tried, we're not getting that much resolution between DMF and NDMA, and hence some of the artifacts that we saw where that ion ratio was changing and where we're seeing a difference in response for a 1 nanogram mil solution dependent on the amount of DMF that we're injecting. We can see that the ultra-core biphenyl, we started to get some resolution. If we're now looking at another or a sort of mixed mode column where we're looking at an ACE2C18AR, we're getting some resolution, and another type of mixed mode separation where we're looking at a mixture of C18 and PFP separations. Again, we're getting a change in that resolution, starting with a very low resolution on the ultra-core C18 and then looking at a much higher resolution with the C18 PFP, and indeed the best resolution coming from the ultra-core biphenyl column. And certainly this is where we're going to put a focus in terms of our future work. So with that, I'll just quickly wrap up what we've gone through. I've talked about the issue associated with nitrosamines, associated with the genotoxicity or specifically the mutagenicity of nitrosamines. Talked about how it was being discovered within the pharmaceutical industry and how the relative interest in this has really risen over the last 2 to 3 years. Talked about the regulatory overview of that, but then focus really looking at development of that analytical assay, looking at starting with the physiochemical properties, how that helps us develop the chromatography, and then looking at some of the considerations that need to be taken when using mass spectrometry, specifically looking at ion suppression effects when we've got valsartan present in the case of NDMA, but also looking at the impact that we would get from potentially residual solvents that still may be left within that formulation, such as DMF. With that, I'd like to thank you for with that, I'd like to kind of just move on to a couple of quick questions, and hopefully you'll be able to answer these. So the first of which is, would you be interested in learning more about Avantor ACE columns? Please click yes or no and then submit if you'd be so kind. And then the next question is, do you feel you've learned something new and about nitrosamine separations or method development? And I sincerely hope that you have learned something a little bit different today, and you can go back and take that into your laboratories and see how that works out when you're developing your own methods, either for nitrosamines or perhaps a wider generation of compounds. And with that, I will finish the presentation, and thank you for your kind attendance and for listening, and I open the floor up to questions. Thank you.
Thanks for the very interesting presentation, Dr. Edge. And thank you, audience members, for submitting questions during the presentation. If you didn't get a chance to submit a question, you still have time. Just feel free to do so, and we'll get to the Q&A portion right now. Dr. Edge, this question came in. Somebody asked, any guidance with regards to intermediate precision or day-to-day variation of the method?
So in terms of the assay that we developed at Avantor, we found that in terms of the response from the mass spectrometer, it was relatively consistent. Obviously, I've shown the impact that ion suppression has. So yes, obviously, if you're working within a pharmaceutical company, you would track the absolute responses as well as the relative responses, and I would strongly recommend the use of a qualifier ion. And hopefully that answers the question in terms of that intermediate precision and day-to-day variation. But certainly the use of qualifier ion will aid that, and from our own experiences, we were finding that the variation was pretty minimal on a day-to-day basis.
Another question that came in asks, is it necessary that the compound, that is the API, be completely soluble for NA analysis?
No. Absolutely not. So indeed, if you look at some of the USP methods, they recommend it's like doing a sort of very simple extraction of the drug if it's possible, so that more focus can be put onto the nitrosamines. The nitrosamine analysis is specifically for the nitrosamine. So if you can remove anything else, then it is actually beneficial. Altering the solubility of your injection solvent can aid the removal of certain drugs, and in fact, that's one of the recommendations that come through from one of the USP methods. But a very good question.
Someone else asks, did you explore any other interfaces that could affect the quantitation?
In terms of that, no. That's a really interesting question, though, because clearly we've looked at two compounds. So we've looked at, like the drug valsartan, and we've looked at DMF, and we were finding that we were getting quite a bit of interference. The work at the moment is very much looking at trying to identify other compounds that could come through the manufacturing process, such as residual solvents, such as some of the impurities that may be found after the drug manufacturing process from manufacturers of certain compounds, to see if they will have an impact. Our experience to date is that pretty much anything that co-elutes with the compound that you're looking at can have an impact in terms of the quantitation. So it's a really good question, and certainly one that I think that is going to be the source of much more work that's coming forward. Thank you.
Here's another one. How important is sample preparation for NA analysis?
In terms of the sample preparation, it really does depend on what you're trying to remove. So, as an example, with the nitrosamines, with the valsartan, we could look at changing the solvent, and that would cause a degree of precipitation with the valsartan. That would remove the bulk of the valsartan. Other compounds, it may not be feasible because of the physiochemical properties associated with that compound. But certainly, the simpler that you can make the sample that you're injecting in terms of number of components, the better the data will come out from the mass spectrometer.
Somebody else wrote in and asked, can Dr. Edge provide the website that provides log D and pKa? Sorry.
Yeah, certainly. So the website is, what I think we might do is just send out an email to the delegates with that slide on. But it's chemicalize.org, and chemicalize is spelled CHEMICALIZ or ZE.org.
That's great. Thank you for spelling that out. I'm sure the attendee appreciates that. Do you have any advice for nitrosamine analysis in protein-based drug products, especially for sample preparation?
So if nitrosamines are expected in protein-based drugs, so I presume the question has been posed by somebody who's perhaps producing monoclonal antibodies, then proteins have a lot of potential for ionization, and so ideally you'd want to try and remove the I'm assuming it's going to be a mAb, a monoclonal antibody. You want to remove the mAb as quickly as possible. In terms of how to remove that, then there are a variety of ways in which you can remove proteins. So a simple form of solid phase extraction may help, even to the point of adding some type of chaotropic reagent like acetylnitrile, guanidine, or an acid to precipitate out the protein. That may also help in terms of the sample preparation. The nice thing is that proteins and nitrosamines are quite different in terms of their size, their shape, and some of the physiochemical properties. So I think SPE might be one. Addition of a chaotropic reagent, so chaotrop is something that changes the nature of the protein, something like acetylnitrile, a strong acid, or guanidine, perhaps even a side exclusion type separation where you use that as a pre-column, perhaps.
someone else asks, in your opinion, how many new nitrosamines will be detected in drugs in the next few years and will be added to regulatory lists?
That's a really good question. I think the audience is coming up with some really very insightful questions. Yeah, there will be more. Absolutely. I think already if you look at the watch list and the compounds that are being monitored in other fields, then there will be other compounds coming through. I think what is prevalent is that when we look at the types and ways in which we monitor those nitrosamines, it's very evident that we haven't captured everything. And so I think absolutely there will be more coming through in the next few years. We've already seen an increase in them. The fact that there's a difference between the number on the EMA and FDA suggests very strongly that even from the FDA we'll be seeing an increase in that hit list of nitrosamines that need to be quantified going forward.
Somebody writes in and says, valstatin concentration is approximately 66 milligrams per milliliters and would be too high for direct injector into the mass detector. Do you use column switch / divide or other sample preparation to minimize this?
It's a good question. So certainly if you were monitoring for the valsartan, you would absolutely, the mass spec is far too sensitive to be running at those concentrations. If you're not monitoring it, then you wouldn't see it. However, the impact in terms of iron suppression would still be there, and because the concentration is relatively high, and that's one of the things that we're looking for. But in terms of what we were doing, in this case, we looked at a simple precipitation method that was given in the USP guidance that I talked about earlier. So yes, absolutely, the individual who gave the question, absolutely right, 66 milligrams per mil. If you're trying to monitor on that on the mass spectrometer, you would probably blow a fuse somewhere down the line. But we weren't trying to monitor it, and we did a very simple precipitation experiment to try and remove the valsartan.
Somebody else asks, how do you explain that suppression is affecting one fragment ion but not the other?
Yeah, it's a really good question. And one of the complexities that we have with mass spectrometers is that we can do some hand waving in terms of the ionization process, but it still is a little bit of hand waving. And when it comes to specific molecules, we know what causes ion suppression. So we know, for example, that during the desolvation process, that coelutin solvents will affect how rapidly that desolvation occurs, and that will depend on the physiochemical properties of the molecule. We also know, for example, that during the ionization, so when ions are actually getting put onto the molecule, that there will be charge competition. So when we talk about the fragment ions and why one fragment ion would be affected but not the other, this raises an interesting discussion because the fragment ion should only be produced within the mass spec, and so consequently we wouldn't anticipate that there would be any suppression coming through because the suppression occurs in the source. So in terms of that, I can't provide an explanation other than we definitely got it from the experimental data. You saw that. So I'm not able to provide an explanation, but I'm happy to have a discussion with somebody over coming up with some theories on that. I think it would be very, very interesting conversations, definitely.
Okay, well, we're coming up at the top of the hour here, so I want to say to Dr. Edge, thank you again for your fascinating presentation, and thank you participants for being a great audience and asking so many great questions. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you On24 for technology and production services, and thank you MacMod for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Jeff Huber. I hope everybody has a great day.